Okay, so yesterday we have discussed about the components of biosensors and the principle of biosensors. In general, biosensors combine both biological transducer and a physical transducer. The biological transducer will recognize biological signal in the form of structure because it recognizes specific molecular structure, for example. Enzyme like only binds to the corresponding product, corresponding substrate in order that it can react, to become, they can react, the substrates so that they can become products. Antibody only binds to the specific antigen, which it recognizes through the molecular structure. DNA also, through its specific sequence, will only bind to the complementary sequence. So, in short, the bioelement of a biosensor recognize specific biological information, either it is structure or biochemical process or other biological or biochemical information. Then, when a reaction occurs, a recognition chemistry occurs, which is in the form of binding reaction or catalytic reaction in the case of enzyme, then some or physicochemical changes will also occur. Physicochemical changes or chemical physics change. For example, heat, there will be heat being either absorbed or being dissipated. Heat being dissipated or being absorbed. Then maybe there will be luminescence, maybe there will be phosphorescence, maybe there will be fluorescence, or changes of absorbance characteristic, or change of mass because of the formation of complex, or transfer of electrons. So all of these usually are in the physical or chemical domain or physicochemical domain. And those changes can be then recognized or transduced using a physicochemical transducer. And yesterday we have explained the type of the bioelement, one that I just mentioned, plus some the larger macro scale bioelement like cell and tissue as well as organ. Then the sensor elements, which can recognize those optical changes or electrical changes or mass change. So this can be, if it were thermal change, then we have to use thermistor or other temperature sensor to recognize the chemical reaction, biochemical recognition reaction. If it were mass change, then we can use, for example, 2cm, quartz crystal microbalance, surface acoustic wave sensor, or also electrical impedance sensor. So based on this, then the most important part, well, I'm not sure if I could say it as the most, but one of the important aspects in the biosensor is how can we couple the action that occurs in the bioelement to be recognized by the physical transducer, by the physicochemical transducer. And here that important component is called as immobilization. Immobilization is the way we immobilize. Normally in common chemistry, the biomolecules are floating in the solution, and they can react three-dimensionally with the corresponding analyte. So then also the recognition will be done also three-dimensionally. Usually then it can only be done using mostly using radiation, or optical domain, like using visual radiation, ultraviolet radiation, or infrared radiation, and we measure the absorbance or transmittance at that particular wavelength. But in biosensor, since we want to take the measurement to the field, Okay, the instrument has to be made small, and also the reaction is made to only occur at the surface of the substrate. So hence the bioelements are being immobilized. We call it immobilized. So they cannot floating around in the liquid in the nanolyte. Instead, they are attached to the surface of the transducer. In that case, then whenever there is a physical chemical change to the bioelement due to the reaction with the analyte, that physical chemical change will immediately also affect the transducer it is attached to. Okay, and that process is called as immobilization, the process of making the bioelement to stick or to be attached to attach to the surface of the transducer. Yeah, why is it called immobilization? Because as the name implies, to immobilize. And normally, the biomolecules is very mobile, floating around here and there by the Brown's law, yeah, Brown's zigzag random movement. Then we immobilize them to standing, yeah, in relatively ordered manner on the surface of the substrate, the surface of the transducer. And to do that, there are 4 different ways uh, to do the immobilization. One is using membrane entrapment. So in this case, we entrap the biomolecule inside a membrane. Okay, inside a membrane, in the porous structure of the membrane, for example, or inside the membrane that it cannot go outside. Yeah. But analyte can go inside to react with the molecule, but then the analyte cannot go outside to the free liquid space. Then second one is using physical adsorption. So as the name implies here, adsorption so means the attachment at the surface of the uh, transducer using physical binding. So physical binding is, for example, van der Waals or electrostatic binding or any other non-chemical binding. So just by letting it dry, yeah, so that it can physically adhere to the surface of the substrate. The third one is what we call as matrix entrapment. So in the matrix entrapment, the material to immobilize the biomolecule is forming like a matrix, yeah, like a porous structure. So in which the bioelement can fill in that porous structure. For example, like in hydrogel, yeah, or not necessarily porous structure, but it can be also attached to uh, that matrix. Yeah, in the case of hydrogel, for example. And the last one is what we call as covalent bonding. So this is very chemistry related method, yeah, where we have to know the chemical structure, the chemical molecular structure of the bioelement, and we modify specifically some part of the bioelement such that then the part can link and can bind chemically covalently to the surface of the transducer. Usually, then it will need a kind of linker molecule, a linker chemical species, yeah, to link between the biomolecules and the substrate. For example, if we have a substrate here, using covalent bonding, for example, we can specifically bind the base part of antibody. This base part, yeah, and then using a special linker to link uh, to the surface of the transducer. Let's say if it were gold, usually we will use sulfur-based chemicals. Yeah, then this will have a certain linker, and by the design of the chemistry, we can make this to be short or to be long. Yeah, and to make it, if it is short, then of course then you will have the antibody more ordered, yeah, orderly arranged on the surface of the transducer. However, then it cannot move around to react or to capture the antigen. So if we design it long, for example, if it was short, yeah, the linker covalent binding like this, so you will have it more ordered, yeah, so like this. But they will not be able to move to free to left and right, yeah, because of the hindrance by the vicinity antibody, by the other antibodies. However, if you put it a little bit long like this, the species, yeah, then they can move around. Yeah. They can move around, and they may capture a more other antigen. So, but of course, then sometimes the time uh, the time it required to capture will be longer compared to the short length antibody. So, using covalent bonding, you can design how your molecule or bioelement will orient it, to which direction it will orient it, to which molecule element of that bioelement attach, which one will be attached to the surface. The drawback is, of course, it is more expensive because it requires additional chemistry, specific chemistry, yeah, and tedious, yeah, tedious and time consuming, and because you have to do these steps of covalent binding more longer, yeah, longer and in multiple steps. The easiest will be to do physical adsorption. However, if we use physical adsorption, yeah, the antibody may not be always like this. Yeah. Sometimes it will be also like this, for example. So the direction of the immobilized antibody will be more random compared to if we uh, immobilize it using covalent binding. So each of these four has advantages and the drawback. Yeah, like this is physical adsorption. It is quick, straightforward. However, the orientation of the antibody will be random. It will be random, and in that case, then reaction also, the reaction side, the sensitive side of the antibody cannot be exploited, all of them. We cannot exploit all of the side. Only some part may be active to bind to the antigen. With matrix or membrane entrapment, yeah, with membrane matrix entrapment, for example, you have a porous matrix like this, and inside your antibody is immobilized, yeah, inside. So it may also still have a random orientation. However, it has relatively spaces between them in order that the analyte can bind to the antibody, even if the orientation is not ordered. This is different with physical absorption, that any, for example, interaction between the antigen and antibody in this case would only happen maybe, most probably only in the site where the reactive site, the Y site, is at the upper part. When it is at this one, the lower part, or even when it is to the side, it may be difficult for antigen to react with that antibody due to the surface interaction. Okay, membrane is similar to matrix entrapment, but usually it has more porous structure. So in membrane, you may have this kind of structure. Yeah, you have the membrane on the, then the bioelement will be entrapped inside this membrane. Okay. Okay, those are the different methods of immobilization. Let's move forward. So yeah, this is the illustration. Yeah, this is a membrane entrapment. We see here a semi-permeable membrane where it has the material, the bioelement is entrapped inside the membrane. B, this is physical adsorption. Okay, when there is a membrane, for example, on top of the sensor, the bioelement is attached to the surface of the membrane here by physical adsorption. C, this is matrix entrapment with the porous encapsulation. So the biomolecule or bioelement is filling the pore structure of that matrix. And finally, covalent bonding, as I mentioned, this is the most ordered immobilization. Yeah, so most efficient in terms of maybe sensing performance. However, in terms of fabrication, it's not that easy. It's tedious and it may be also expensive. So in the membrane entrapment, a semi-permeable membrane separates the analyte and the bio element. The sensor is attached to the bio element. Okay. And in the semi-permeable membrane, in the membrane entrapment scheme. Then in the physical adsorption, it depends on the van der Waals forces, hydrophobic forces sometimes, hydrogen bonds, and ionic forces to attach the biomaterial to the surface of the sensor. With matrix entrapment, we have porous encapsulation matrix around the biological material to help it binding to the sensor. Here, while in the covalent bonding, the sensor is treated as a reactive group to which the biological material can bind. So this reactive group is the one that we need usually have to be specific to for particular surface and for particular biomolecule. That is why it is not that not that cheap to develop a covalent bonding immobilization. Okay, so why do we use biosensor? As I have maybe mentioned in previously, the most significant important properties in biosensor is due to since it has this bioelement, and therefore it has the specificity and sensitivity towards the target analyte. Okay, so biosensor has the specificity and selectivity and sensitivity. Specificity or selectivity is because they are so selective towards certain molecule, they will not bind to other molecule. They only bind to the specific molecule they are designed to. Antibody that towards SARS-CoV-2 virus will only bind to SARS-CoV-2 virus and cannot bind to other virus or other protein. Similarly with others, enzyme glucose oxidizer only oxidize glucose and will not oxidize, for example, fructose or lactose, even though they are of the same chemical family. Okay, so these are that sensitivity is the one that being purchased in the biosensor implementation. Then sensitivity, they are sensitive because instead of the molecule floating around in 3 dimensions in the liquid, the biomolecule is attached to a surface. So in that case, then the reaction happens on the surface. And at the moment where the recognition reaction occurs between the bioelement and the corresponding analyte, then that physicochemical change can be directly recognized or detected or measured by the transducer, because the bioelement is attached to the transducer using the 4 immobilization methods that we have just discussed. Okay? The specificity of biosensor is entirely governed by the properties of the biological component, where the analyte is interacting with the sensor. Then the specificity, and therefore enzyme will only recognize its substrate. Antibody also will only recognize its antigen. Okay, then sensitivity on the other hand, especially demand chemical modification, enzyme will with far higher affinity than operating on the chemically similar molecules. In the case of other small sugar in the glucose detection can elicit a small amount of cross-reactivity if present in sufficiently high concentration. Only if present in sufficiently high concentration. When they are not that high concentration, then the glucose oxidase, for example, in glucose reduction will only recognize glucose. Now here, however, this is mentioned in this book that the disadvantage is the inherent instability, yeah, of biomolecules. This is what is one of the problem in biosensor, because we use enzyme, for example, yeah, enzyme and also antibody. They are proteins, and proteins denature eventually with temperature, with storing condition, dependent on the buffer and humidity, etc. Okay, that's why many strategies have been deployed to restrain or modify the structure of biological receptor to enhance their longevity, to enhance their lifetime. Yeah. Therefore, if we purchase, for example, glucose sensor, if you purchase in any apotheke, yeah, in any pharmacy store, usually the permanent part is actually the electronics. Okay, the strip, yeah, the element where the enzyme, the God, is being immobilized to recognize the glucose is usually provided as strips, and the strips is one-time use. Yeah, after you use it, it cannot be used anymore. You have to dispose it, and they have also expiration date, yeah, because of the enzyme condition, as well as for many other kits, yeah, in the medical world that rely on the use of bioelement. Okay, so far, any questions regarding this? Okay, we have explained actually this one yesterday, but we can review again one by one. So the common bioreceptor component are classified into 5 groups. Okay, the first is enzyme. Yeah, these are biological catalysts basically. Yeah, proteins that catalyze specific chemical reaction. It can be used in purified form. Yeah, of course, then it will be more expensive, like glucose oxidase, what else? Lactate oxidase, many other enzymes, or be present in a microorganism or in a slice of intact tissue. Now, this is one way to make it cheaper. Is you don't have to purify it from bacteria or from a tissue, yeah, because maybe it is too difficult and too tedious, and it requires special column to purify it. But instead, you can just take the microorganism, bacteria that has specific enzyme, or tissue that has specific enzyme, and use it as is, or as a bioelement in a biosensor. But of course, it comes with a catch. Yeah, there is a drawback in using such an approach. The drawback will be that then, since in the whole bacteria cell, or for example, a whole intact tissue, the structure is more complex than a single molecule, then it will take time for the analyte in the sample solution, yeah, to interact, to go through the structure of that bacteria or the tissue, to find the enzyme and then react with the enzyme. Okay, so this will slow the response time of that sensor. So this is the drawback. Of course, it is cheaper compared to if you want to use pure enzyme. Okay. And however, the advantage also of using microorganism like the whole bacteria or intact tissue is that it will have longer time life, yeah, shelf life, yeah, compared to if you just use the single purified enzyme, because then it means if you use the bacteria or whole microorganism or an intact tissue, then the enzyme is preserved in its natural environment, albeit in very limited, confined environment. So in terms of the humidity, pH, etc., it is still maintained in that small microenvironment. So in that case, then the enzymes there may have a longer lifetime. Okay, so those are the drawbacks and advantage and disadvantage of using pure enzyme, or when you have to choose pure enzyme or microorganism. In our case, for example, in developing world, in developing country, we still find it difficult. It is still difficult for us to purchase pure enzyme. Even only common enzyme that can be purchased at relatively low cost, like glucose oxidase, lactate oxidase. But very specific enzyme usually still are expensive. In that case, then it will be easier, it will be cheaper that if we can produce that enzyme in microorganism, in a bacteria. For example, using genetic engineering, you can transfect the vector of E. coli to produce that enzyme. And no need further purification. We just use the E. coli cell as it is, use it as the bioelement of the biosensor. Second category is the antibodies and antigen. So an antigen is a molecule that triggers the immune response of an organism to produce an antibody. An antibody basically is a glycoprotein produced by the lymphocyte B cells, which will in mammals, which will specifically recognize the antigen that stimulated its production. Okay, so this is also with antibodies and antigen, we can have either polyclonal antibody or monoclonal antibody. If in polyclonal antibody, then the antibody that is produced is not sensitive towards only one side of the antigen. Okay. So let's say, for example, you have this structure, the structure of antigen. Okay. Then if you put into, you could produce different, if you put inside mammals, inside rabbit, or inside mice to produce the antibody of this. The antibody may be sensitive towards this region of the molecule. It may also sensitive towards this region. Other may be sensitive towards this region. Then this is called as polyclonal antibodies. So you have in that production, after from the blood of that mammals or rabbit, you purify the antibody part. You will have these basically three different types of antibodies that can bind to that antigen, but at different side of the antigen, the polyclonal. So if you want to make it specific, more specific usually, then you go for monoclonal antibody. Monoclonal antibody, then you purify further. So you only, for example, you want only to get this antibody that bind only to the specific site of the antigen. So this is called as the monoclonal antibody. In recent years, however, people sometimes do not use animals to produce the antibodies. They use instead cell culture to do antibody engineering, they call it. Although it is not yet common, or it is a bit expensive. Okay. Then further, the third one is nucleic acids. This is the recognition process which is based on the complementary nature of the base pair. Okay, of the base pair. Adenine, thymine, and cytosine and guanine. So adenine will bind with thymine and cytosine will bind with guanine in the DNA, in the double helix of DNA. And the sensors are also usually called as the genosensor. As genosensor. Alternatively, interaction of small pollutants with DNA can generate the recognition signal. So it's not necessarily that you use a nucleic acid to detect other nucleic acid, but also sometimes that nucleic acid can detect also small pollutants. In recent year, we also know what we call as a new types of molecule, recognition molecule called as aptamer. What is aptamers? Aptamers is a series of nucleic acids which can make a secondary and tertiary structure and then will function like an antibody. So this is also one of the cheaper way to develop a molecular binding biological element, which does not necessarily use animal to produce it. So in natural antibody, you need to use rabbit or mice. You inject the antigen that you want to detect as biosensor, and then you produce the polyclonal or monoclonal antibody, and you use it as the biomolecule element for your biosensor. However, nowadays people also instead use series of DNA, A, G, C, etc., etc., double strand, and then this DNA will make a secondary or tertiary structure, sometimes like this, very complex structure, and as a whole molecule, they will behave like a antibody that will bind to specific molecule, to specific antigen. And then how can we find the correct aptamer to bind to specific antigen? And that process is called as combinatorial chemistry. Combinatorial chemistry. So usually, until now, they design it randomly. For example, we design an aptamer consisting of 100 base pair. 100 base pair. BP is a base pair here, means one single sequence series here. So this will be like this. This is base pair. So this one, 1, 2, 3, 4, 5. This is 5 base pair. So for example, within 100 base pair, they randomize the sequences. And they test the antigen to this different pool of randomized sequence of aptamers. And then from there, they will look which one will form a binding reaction. Okay. So then they will pick, then they will save that sequence to produce further, using a process of combinatorial chemistry. And it is cheaper. And with aptamers, with nucleic acid, it is also has longer lifetime compared to antibody, because antibody is a protein, and protein easily denaturates when it is put out of its natural environment. While aptamers, nucleic acid usually can stay active even outside, as we can see in virus. Even virus, the protein, the DNA inside, when it is not touched to anything, it can still bind, something like that. And that is why when you want to disinfect from virus, you get rid of the encapsulation protein, which is usually made from membrane, lipid membrane, by using soap. Okay, then the third one is the fourth one, is cellular structure. Cellular structure or whole cells, the whole microorganism or a specific cellular component receptor. For example, a non-catalytic receptor protein, ion channels, and lipid membrane is used as the biorecognition element. So whole cell. So actually, in previous example, I have mentioned also whole cell of bacteria, for example, can be used as the bioelement. But not only bacteria, even we can use also mammalian cells as a biological recognition element. Yes, this is when do we use cellular structure of cell? When we want to look at the overall cellular response rather than in particular recognition element response. For example, we would like to see the toxicity of certain classes of chemicals towards neural cells, for example, neural cell. Then what we will put the neural cell as the bioelement, and we will drop the different types of the chemicals, and then see their interaction. Then we can measure using the physical transducer can be electrode, for example, and then we can measure the potential change due to the interaction of the neural cell with the particular chemicals. So this is when we want to see toxicity, the neurotoxicity. And this can be applied also to many other types of cells. And finally, the fifth type of bioreceptor component is the biomimetic receptor. And this is usually not exactly biological molecule, but instead they are genetically engineered molecule or artificial membranes or molecularly imprinted polymer, MIP. Actually, molecular imprinted polymer is made from polymer, but it can behave like an antibody that can bind to specific molecule. And how can we make that? Also using the process similar like what I have explained about aptamer. But the difference is with aptamer you use nucleic acids. With molecular imprinted polymer you use different types of polymer and make them, and then to have specific structure that can bind to specific molecule. So the MIP can mimic a bioreceptor or ion channel and something another. So in recent days also other biomimetic receptor, I have mentioned maybe earlier yesterday, what we call as Metallo organic framework. Metal organic framework. This is just in recent years, maybe in the last 5 years to 10 years. So this is usually when we want to have something that mimic catalytic property of enzyme. So if you Google now, Google Scholar, for example, non-enzymatic glucose sensor, you will see many of them are made using MOF, metal organic framework. These are made from non-organic material, usually metal oxide, like copper oxide, tin oxide. So similar material like the gas sensor. However, they are engineered to have a more complex three-dimensional structure, not just the usual crystalline structure, but more supramolecular structure. So that then they can have their porous structure, their binding site will mimic the structure of enzyme. This is something which is in recent years, people are developing. Okay. So basically, biosensors are typically classified by the above of recognition element or transduction element. A sensor might be described as catalytic biosensor if its recognition element comprised of an enzyme or series of enzymes, or a living tissue or whole cells derived from microorganisms such as bacteria, fungi, or yeast. However, the sensor might be described as bioaffinity. So those 5 are the main classification of bio recognition element, but the type of biosensor itself, 2. The 2 main classes. The first is catalytic biosensor, the other one is bioaffinity sensor. So in catalytic biosensor, the principle is based on catalysis reaction, catalyze a reaction, while bioaffinity is based on the binding between the recognition element and the analyte to form a bispecific complex formation. Accordingly, the reaction of antibody with antigen or hapten or the ligand with the receptor would be employed. Okay, so that will be all for today. Next week we will discuss in more detail each of this type of catalytic biosensor and bioaffinity sensors, and further also some explanation on the physical transducer used as the secondary transducer to measure or to detect the physicochemical changes when recognition or catalytic reaction occurs. Any questions so far?
No, sir.
No questions?
Sir, itu yang di bagian yang di NA yang ada yang pollution itu maksudnya pollution gimana ya?
Yeah, for example, DNA are sensitive also towards heavy metal. Why? There is electrostatic binding in the DNA double strand. So if you are introducing some heavy metal, for example, this will disrupt the binding between the molecular structure. Otherwise, like that. Otherwise, it is similar like the aptamer mechanism. So the DNA having DNA structure having a secondary or tertiary structure. You understand what secondary and tertiary structure means? Yeah. Primary structure is the sequence, the AGTC sequence. Secondary, if it makes a loop, for example, a loop, or tertiary, if it makes another more complex structure. In protein, it can go to until quaternary structure. So this structure, as I mentioned, with aptamer, for example, can bind to specific molecules. Yeah, either non-organic molecule like metals, ions, for example, or organic molecules. Yeah. So this also then they can bind and then it generate recognition signal in the form of mass change or in the form of perhaps transfer of charge, something like that. Okay.
Thank you, sir.
Please Google Scholar for further detail. Yeah, it is not also mentioned here in the textbook in detail. And I use DNA as sensor, but for protein, yeah, during my master degree study, but not as small pollutants. So also I'm not that familiar also, but my guess is like that. So either it is its interaction with heavy metals, yeah, or metals that can disrupt its binding. Yeah, therefore, then it generates mass change, or it functions as like an aptamer. So similar like an antibody, but then specific to a certain molecule. Okay, please have a read on aptamer, MIP, MOF. Yeah, these are new bioelements. Yeah, maybe not in this textbook also. This textbook is written by the Chinese professor in the first decade of the 21st century. So far until now, the field has developed quite significantly, yeah, with different new bioelement. Other questions, maybe? If not, then that will be all for today.